Why is this not? Okay. I think I'm having a bit of a technical problem here with my computer. In any case, I'm going to talk today about cell-based neurotransmitter fluorescent engineered reporters, or sniffers, as we like to call them. So a brief overview of how sniffers are constructed. We take a receptor, a G protein coupled receptor of choice, your favorite G protein coupled receptor, and we stably transfect this in an HEK. In addition to that, we also stably transfect in a FRET calcium sensor, which will emit two wavelengths, YFP and CFP. Being able to image both wavelengths simultaneously actually allows you to suss out movement artifacts from true actual fluorescence due to calcium binding to the FRET protein and causing change in configuration. We have sniffers that have been designed to detect acetylcholine, dopamine, norepinephrine, orexin, VIP, and serotonin. Just a quick overview of how sniffers compare to other methods, other popular methods. With respect to microdialysis or cyclic voltammetry, sniffers offer a superior sensitivity, temporal resolution, spatial resolution, and most importantly, it allows you to visualize different types of neurotransmitters simultaneously. So this is an in vitro response profile of a particular sniffer, an alpha-1 sniffer. When you image the two channels simultaneously, you get a drop in emission in one fluorophore versus an increase in emission in the other, and this occurs as a result of binding to neurotransmitter and activating a signaling pathway that results in an increase in calcium concentration intracellularly. As you can see, in terms of temporal resolution, when you put the sniffers in a well where you can flux on neurotransmitters, upon delivery of the neurotransmitter within 5 seconds, the sniffers, you can detect an increase in FRET ratio that lasts well over for about 30 seconds. And this is just an example of what the sniffers look like when you implant them, when you do an in vivo implantation of them. It's pretty easy. You basically take a glass pipette, insert it into different regions. This is a cortical region, and deliver the sniffers, and you can pretty much image them the same day during various behaviors. So this is just an overview of what has been done in David's lab previously. We wanted to see whether or not we can detect an increase in dopamine levels in the prefrontal cortex as a result of classical conditioning. Classical conditioning, I'll remind you, is when you take a stimulus like a tone and pair it with an unconditioned stimulus, which is a reward. And as Arno had discovered in this paper, basically with training, you can see that the dopamine release actually gets closer and closer to the unconditioned stimulus. And this coincides well with previous reports showing that in the midbrain, where these neurons are actually located, there's an increase in firing to the conditioned stimulus as the animal learns that conditioned stimulus becomes paired with the reward. And basically the learning, when you look at the rate of change in licking onset time versus the rate of change in dopamine onset time, you can see that there's a linear relationship between learning and dopamine release. So we covered cortex, and what I want to discuss with you is, or talk to you about, or present to you is what about other brain regions, those that are located under the cortex, such as hippocampus. So in collaboration, with Darwin Berg's lab, I developed this method in David's lab, and this is a way to chronically image the hippocampus utilizing the 2-photon setup. And so what we have here, I'll go slow through this because I don't know how many people have ever seen this thing before. But what we have here is a titanium implant, which is 8 millimeters outer diameter, 3 millimeters inner diameter, and projects exactly 1.5 millimeters down onto hippocampus. We have to do some suctioning of the overlying cortex, but basically that's just to allow us to fit the implant in and dental cement it onto the animal. And this is how the setup works. Basically, the animal is placed under the 2-photon scope. In this previous project, I looked at how nicotine acute nicotine administration affects hippocampal activity, and this is just an example of what we can see, hopefully. So it's clear that we can see through hippocampus repeatedly. And before I go on, I want to discuss some of the future projects we have with the sniffers and hippocampus. Celine's going to continue on in the cortex. She's going to look at acetylcholine release and neurovascular dilation during sleep. Conrad, Fu, and I will look at atypical dopamine release by norepinephrine terminals in hippocampus, as well as blood flow and neuropeptide release in cortex, and reinforcement learning-based dopamine release in hippocampus and striatum. So, with respect to goal number 2 here, we're doing this in collaboration with Richard Morris's lab. And what Richard Morris had shown in a previous, in a Nature paper, was that, and this is what I mean by atypical dopamine release, is that when you label the dopaminergic neurons in VTA in midbrain and look at the terminals in hippocampus, you can see that the dorsal hippocampus, CA1, gets very few dopaminergic terminals. But on the other hand, if you label noradrenergic neurons in locus coeruleus, you can see that the hippocampus is populated by a lot of terminals from locus coeruleus. Richard Morris's contention is, and this is in the Nature paper if you want to see it, is that dopamine, these terminals can actually release dopamine, and they do this to mark salient events during periods of episodic memory. For example, so episodic, basically you remember your memories are remembered in episodes. If you do something every day, like put cream cheese on your bagel, you're going to pretty much forget about 90% of that. I mean, if that's something you do daily. But if a pink elephant passed by you, you're gonna remember that piece of memory because it's actually tagged. And Richard Morris's idea is that tagging is done by a release of dopamine. So this is something we've recently done, and that is to actually inject sniffers into hippocampus and put the implant on, and show you some preliminary data showing that actually there is actually some type of dopamine release in this dorsal hippocampus, and we don't know exactly what it's about. We have the fret response here and relative to the velocity of our wheel. So this animal is running on a treadmill, and as you can see, it's not really, doesn't really coincide with running or movement of the animal. Okay, in any case, so new sniffer technology, as I mentioned, we were starting to get into neuropeptide sniffers. These are brand new sniffers we just got a few days ago. One sniffer detects VIP neuropeptide. The other one detects somatostatin. This is the dose response curve of the VIP sniffer, and again the dose response curve of the somatostatin, and it's relative for the VIP sniffer. This is the relative affinity of it to other neurotransmitters as well as VIP itself, and somatostatin the same here. One last thing I want to add. So for the most part, in the past, we've basically just injected the sniffers. I'm a molecular neuroscientist. I thought that I could dress these cells up even more. There is actually evidence of that in the literature. You can actually transfect these HEKs with neuroligin 1AB to give them higher affinity both to astrocytes and to neurons. And so that's another future direction that we'll look into. The sniffers are also all one color. They either emit two colors from it. They emit CFP and YFP. I'll also introduce a different color to them, either blue fluorescent protein or TD tomato, so that we can actually distinguish visually while we're imaging the two different populations of snippers. And with that, I'll take questions.